Welcome, everyone. Today, I'll be presenting a summary and discussion of a significant research paper titled Deciphering Cancer Stemness in Tumors, Novel Stemness Index for Oncogenic Dedifferentiation. This study was conducted by researchers who aim to understand the role of stemness in cancer, progression by developing new stemness indices. The findings have important implications for cancer biology and potential therapeutic approaches. When comparing primary tumors to metastases, metastatic samples typically exhibited higher stemness indices, suggesting that dedifferentiation plays a role in tumor spread. Single-cell analyses revealed significant intratumor heterogeneity in stemness, particularly in gliomas. This heterogeneity may contribute to challenges in treatment, as subpopulations of cancer stem cells can drive resistance and recurrence. The study found that tumors with high stemness indices often have fewer immune cells infiltrating the tumor microenvironment and lower expression of PD-L1. This suggests that these tumors might evade immune detection and could be less responsive to immunotherapies that rely on activating immune cells. Understanding the relationship between stemness and the immune response is crucial for developing effective treatment strategies. By using CMAP analysis, the researchers identified several compounds that may inhibit cancer stemness. Notably, some of these compounds are already used clinically for other indications, such as dopamine receptor antagonists. This opens the possibility of repurposing existing drugs to target cancer stem cells, potentially improving therapeutic outcomes. The shared mechanisms of action among these compounds highlight key pathways that could be targeted in anti-stemness therapies. In summary, this study successfully developed novel stemness indices that provide valuable insights into the degree of dedifferentiation in tumors. The indices correlate with important clinical features, including tumor aggressiveness and patient outcomes. By understanding the role of stemness in cancer progression and therapy resistance, we can improve prognostic tools and develop targeted treatments that may overcome current therapeutic challenges. Looking ahead, it's essential to validate these stemness indices in clinical settings to confirm their utility as prognostic biomarkers. Further research should focus on testing the identified compounds in preclinical models to assess their effectiveness in targeting cancer stem cells. Combining stemness targeting therapies with immunotherapies could enhance treatment responses. Ultimately, incorporating stemness assessments into personalized medicine approaches may improve patient outcomes by tailoring therapies to the dedifferentiation status of tumors. I want to acknowledge the authors of the original study for their significant contributions to cancer research. Their innovative approach to quantifying cancer stemness has provided valuable insights that could impact future therapies. I also recognize the importance of TCGA and PCBC for providing the extensive datasets used in this research. Thank you all for your attention, and I'm happy to take any questions. I'd now like to open the floor for any questions or discussions you might have regarding this presentation. Please feel free to ask about any aspects of the study, its implications, or areas that you'd like me to elaborate on further. Thank you for your time and engagement during this presentation. I appreciate your interest and the opportunity to discuss this important research with you all. In this slide, we'll explore the process of extracting information from unstructured text. We will cover the techniques, tools, and goals associated with this important task in natural language processing. To begin, it's important to understand the concept of stemness in the context of cancer. Stemness is the property of a cell that allows it to self-renew and differentiate into various cell types. During cancer progression, cells often lose their differentiated features and acquire stem cell-like characteristics, leading to more aggressive and treatment-resistant tumors. The tumor microenvironment plays a crucial role in this process by providing signals that can promote or inhibit stemness. The central question addressed by this paper is whether novel stemness indices can be used to quantify the degree of dedifferentiation in tumors and uncover underlying biological mechanisms. The researchers hypothesized that these indices, based on gene expression and DNA methylation data, could provide insights into cancer stemness and its associations with various molecular and clinical features. The researchers utilized publicly available datasets, including profiles from stem cells and their differentiated progeny, as well as extensive data from TCGA. They developed two stemness indices using the OCLR algorithm, one based on mRNA expression, mRNA-ASI, and the other on DNA methylation, mDNA-ASI. These indices range from 0 low stemness to 1 high stemness. The indices were validated using external datasets and were applied to TCGA samples to explore associations with various molecular and clinical features. The mRNA-based stemness index, mRNA-AC, effectively distinguished stem cells from differentiated cells. In tumors, higher MR and NSE values were observed in types known for their dedifferentiated phenotypes, such as germ cell tumors and basal breast cancer. The index correlated positively with known stemness genes like MYC, OCT4, and SOX2, and negatively with EMT markers. This suggests that tumors with high mRNA maintain stem cell-like characteristics and an epithelial phenotype. The DNA methylation-based stemness index, MDNASI, was developed by focusing on hypomethylated enhancer regions specific to stem cells. The index effectively differentiated stem cells from their differentiated counterparts. In tumors, high MD and ASI values were associated with hypomethylation at enhancers critical for maintaining pluripotency, such as those bound by SOX2 and OCT4. This indicates that DNA methylation patterns play a significant role in regulating cancer stemness. While mRNA-AC and MDNA-AC generally showed positive correlation across various tumor types, exceptions were found in gliomas and certain other cancers. In gliomas, IDH1/2 mutations cause widespread DNA hypermethylation, leading to lower MDNA-AC despite high stemness indicated by mRNA-AC. This suggests that mRNA-AC and MDNA-AC capture different aspects of cancer stemness, and both indices are valuable for a comprehensive understanding. Applying the stemness indices to specific cancers demonstrated their ability to stratify tumors based on dedifferentiation. In breast cancer, high mRNA-AC was associated with the basal subtype, known for its aggressiveness and poor prognosis. In AML, the index correlated well with the degree of myeloid differentiation. In gliomas, high MDN-AC was linked to high-grade tumors and adverse clinical outcomes. Across various cancers, the stemness indices revealed associations with key mutations, microRNA, and protein expression levels. For example, mutations in genes regulating epigenetics were linked to higher stemness, suggesting a role in dedifferentiation. High stemness was often correlated with higher tumor grades and worse clinical outcomes. Additionally, tumors with high stemness indices tended to have lower immune cell infiltration and PD-L1 expression, which may impact responses to immunotherapy.